Hi, everyone. In this lesson, we're going to go over chapter 13.1 of your textbook, which is between pages 206 and 207, which covers Mendelian genetics. So this page in particular, you do not have to copy down into your notes, but I want to introduce you to basically the father of modern genetics, Gregor Mendel. Now, Gregor Mendel was a monk from the 1800s, and during his lifetime, he wasn't really well known, but he liked to study pea plants a lot, and he did all these experiments on pea plants and things like that. And he published this, at the time, little known book that basically recorded all of the information from his experiments and what he observed and what he thought was happening. And it turned out when his book was later discovered in the 1900s, all this information basically explained why certain things occurred, why certain things were passed on the way they were. They basically helped make evolution make more sense when humanity discovered evolution. And this is all the more impressive because he did all these observations and predicted correctly what was going on without even knowing what DNA was. Now, his experiment basically used a lot of pea plants. Now, there's a lot of advantages of using pea plants. One, their genetics is very simple, so the traits he observed were much easier to understand. We'll learn later on some more complicated way genetics can occur, but he luckily picked a species where it was very straightforward. Another thing that's kind of interesting about pea plants is they have male and female parts, and they are diploid, so it's going to track a lot of the ways traits are passed on in humans. But because they have male and female parts, they are doing sexual reproduction. So even though they can self-fertilize, you can still breed them with other plants. And it's not asexual. It's still sexual because what's happening is the flower is producing male sperm and female eggs and joining those sperm and eggs up randomly. So the kids, if the parent has mixed genes, the kids will actually end up still being different, even with a self-fertilized plant. But you can also breed one very different type of pea plant with another very different pea plant together and observe the child. Okay, so these experiments involved controlled breeding, where he basically took two parents with very clear different traits or same traits and bred them together and then raised the seeds up and observed the offspring and wrote down his conclusion. And he could also very easily breed the offspring together and then track what went on with them. All right. Now, because he didn't know what genetics was at the time, because he was the founding father of it and he didn't even know about DNA, he actually started with what was known as true breeding plants because he wasn't sure what was going on 100%, but he recognized that certain plants consistently always, if you just self-fertilize them, would consistently always produce only one type of trait. So if you're tracking a flower color, it would always, no matter what, with every generation, always produce white flowers. We now know this to be something called homozygous, meaning you have all the copies of alleles of information for that gene only produces white flower protein. But he didn't know that at the time. And that when he crossed two plants with different traits, he could observe what the hybrid trait would look like, and they would actually often be the ratio of the hybrid traits or these cross or any cross would actually be fairly predictive. And then basically he realized that information passed on, hereditary information that you pass on from parent to child, is passed on to discrete units. Now he didn't have a word for it, but that basically is genes. A chunk of information is being handed from the parent to the child in a way that is consistent and predictable. Okay? So traits come in alternative versions. All right? So traits, if you forgot what trait is, it's what you see and observe in the individual. And these alternative versions of a trait is what we call an allele. It's a variation in nucleotide sequence at a locus or site of a gene. Now, all those words are very confusing, so let me explain it a little bit better. So, organisms have genes. That's the information for protein, and genes are always consistently found in one location or locus on a chromosome. Okay? If you are diploid, which is what humans are and these pea plants are, you have two copies of each chromosome, which means that for any given gene, you have two different versions of that same gene. Okay? Or two copies of that same gene. An allele is basically the specific recipe at that gene. So the gene for eye color is in this part of the chromosomes, but the alleles meaning the type of gene, can come in different flavors, meaning the specific information that leads to purple flowers versus the specific information that leads to white flowers. So again, this gets a little bit confusing, so let me define it a little bit further. An allele is a variation on a gene. A gene is a specific part of your chromosome that provides the info for a protein that's going to lead to what you observe, the trait. Okay? So the gene is the information your body uses to make eye color. The allele is whether the specific information at that gene leads to a brown eye protein or a blue eye protein. Okay? Sometimes the word gene is used interchangeably with locus because locus is the location of the gene and it's always consistent for that gene. So sometimes instead of saying gene, we'll say the eye color locus indicates that you have a blue color. It just means gene. Okay, so what did Mendel's findings mean? Well, he realized that a lot of people at the time assumed if you are tall and your child is short, if you mix them together, you'll always get a medium trait, some kind of blended trait. But that was not always the case. That was sometimes the case, but not always. So for example, he realized that if he mixed purple and white pea flowers together, you actually didn't get a light purple color, a mix of white and purple. You actually ended up with purple flowers, which meant there were sometimes cases where an allele masked another allele. Purple masked white. So if you had a purple flower mixed with the white flower and you had that kid have one purple flower gene and one white flower gene, one of them was dominant. The purple flower gene, or allele, sorry, the purple flower allele masked the white flower allele. And oftentimes that was because the white flower allele had an absence of a functional protein pigment, while the purple flower allele did not have that. Oftentimes the dominant allele just means that you make a protein that's either hardier or stronger or even just present, while the one that doesn't show up is either just weaker or it may not even have a functioning protein. That's not always the case, but that's often the case. Now, we're going to learn how this gets used later, but sometimes we represent the dominant allele using uppercase letters. So you might be familiar with Punnett squares or genotypes and the way they're written out from middle school, but we're going to sometimes use letters to represent the DNA, big letters versus small letters. All right? Recessive alleles only appear when there are no dominant alleles. So a recessive trait only appears if both copies that you receive from the parent are recessive types of alleles and not dominant. So the only way a white flower gene would show or allele would show up in the kid would be if both parents passed on a white flower allele for that flower color gene. And we often represent this with a lowercase letter. In this case, lowercase a, but you can use any letter. Just for any gene, you want to use the same letter. So maybe you use F, uppercase F, lowercase f, something like that for a gene. Okay, now genotype is the specific allele combo that's often represented with that mix of letters, where we're using uppercase letters to indicate a dominant allele and lowercase letters to indicate a recessive allele. So it's a particular set of alleles an individual carries for a trait. Each allele is represented by a single color. So for the purple flower and white flower here, the genotype is big P for some purple flowers, or maybe big P, little p. That's genotype, to indicate it has one copy of the purple flower allele and one copy of the white flower allele, or possibly two copies of the purple flower allele. Or for the white flower, two copies of the white flower allele. And we know it's two copies because to show the white flower or a recessive allele, you have to have only that recessive allele. The phenotype is the actual trait, the actual outcome. So whether you have purple or white flowers. Sometimes if you know the phenotype, you'll know the allele genotype. Not always, like the white flower one, because the only way to see a recessive allele is by having it be homozygous for that white flower allele. But other times that's not the case, especially for dominant traits. So like seeing a purple flower, You can't tell exactly whether that means it has two copies of the purple flower allele or one copy of it and a different copy for the other one. So we sometimes use the terms homozygous and heterozygous to indicate the allele blend. Okay? So in this case, homozygous, also used interchangeably with the word true breeding, because every generation in theory would always consistently get the same alleles, mean that both copies that this one parent or one individual has is the same type of allele. Two purple flower genes, two white flower genes, two tall genes, two short genes, whatever type of gene you happen to be looking at. Other times we use the term heterozygous, which means hybrid. So hybrid means like you blended two things together. Hybrid was a term that because he didn't know about genes at the time that Mendel used in his stuff. But this hybrid organism then has two different alleles for their gene. So in this case, one purple flower allele and one white flower allele, one tall allele and one short allele, whatever you're observing for this one gene. All right, so this is a lot of vocabulary. So basically, you just need to understand the vocabulary in particular and with how it's used. The next lesson has a lot less vocabulary, but we're going to use these vocabulary words and apply them to actual problem solving and prediction. Okay? So in this case, we just have our general takeaways over here, which explains why Mendel's experiments were very important, and then just a little bit background on how these terms are used within context. And these were the vocabulary words that you will really need to understand very well for this unit. That concludes the lesson. Bye.